Hey, Miss Shannon, sorry that took us a minute. We're scrambling from lab. You know how it is.
Hi, Shannon.
Okay. If it opens, there we go. All right, so this is some fun stuff today. It's some really interesting stuff. This is cool material to wrap up our genetics unit. So you guys have a test this week, right? We're leaving unit 4. We're moving to unit 5. Unit 5 is one of my favorite ones. Has some of my favorite labs in it. Actually, it has my all-time favorite lab in it. I love it. I love it. I love it.
The frog.
No, not the frog. Although I do love all those. Now, as far as I guess my favorite non-dissection lab, let me do it that way. No, it's really fun. I enjoy it. Some of you guys have done something similar in the past, so don't spoil it for newbies. All right, so we are doing genetics and genetic engineering today, and this just kind of brings everything back full circle where we started, which is really kind of good because there's been a lot of nitty gritty in genetics, right? We get down to the molecular level and then sometimes you forget the big picture. So it's smart to kind of circle back around and look at where we started with things. All right, so genetic engineering. This is a fun picture I found. By cloning cells and modifying genes, scientists in Korea have developed, hey guys, can you keep it down? Thank you. Scientists in Korea have developed cats that glow bright red in the dark. Oh, I knew some eyes would come up when I said that. Same eyes I expected to come up. That's fun. So the cloned Turkish Angola on the left has a fluorescent protein in its skin cells. The protein gives off a red glow when exposed to ultraviolet light. And the ordinary Turkish Angola on the right lacks the red fluorescent protein, so it appears green under ultraviolet light. Isn't that interesting? Now, why in the world would we want to make a cat glow? Well, there's reasons, actually.
Yeah.
There are reasons.
For fun.
Not just for fun, although it is pretty cool, I will say. So what's selective breeding? So we're going to talk about genetic engineering here from the standpoint of molecular work that's done, as well as large scale stuff that we've been doing for millennia. So as humans, we've been doing selective breeding for millennia. So this is when we breed organisms to try and get the desired characteristics into the next generation, okay? So we do this with everything from animals to plants to, plants we use for industrial purposes, not just for food. So we're talking about things like horses and cows and plants and, oh, is he all right there? Oh, he stole something. So humans bred dogs for thousands of years to produce better hunters, retrievers, companions. Then we have allowing only those animals with wanted characteristics to reproduce into the next generation. So if you have a dog that's a really good hunter, and you've got a male dog that's a good hunter and a female dog that's a good hunter, and you want more good hunters, those are the two you're going to breed to make more good hunters. This is how we've gone from the wolf to nearly 200 varieties of the species of dog.
Chihuahua.
Yeah, Chihuahua. Yeah, that's a great one, isn't it? So humans use selective breeding, which takes advantage of naturally occurring genetic variation to pass wanted traits on to the next generation of organisms. Say there was an overlord who wanted good piano players out of our classroom, and they came in and they were like, I only want people with big hand spans. Raise your hand if you would be out. I would be out. Yeah, and those of you with the big hand spans would be bred into the next generation. But we did notice that most of our largest hand spans belong to male students. So a smart overlord with selective breeding would come in and say, well, I need my females with my biggest hand spans and my males with my biggest hand spans. And that's how I'll get great piano players in my next generation of big hand spanned humans. Yeah, I'll make that into a word. So produces new varieties of cultivated plants and all domestic animals, horses, cows, cats, selective breeding for particular traits. And then also Native Americans were doing this too. So they bred teosinte, I think is how they say it. It could be teosinte, I don't know. It's a wild grass. And they basically bred it into what we consider corn, right? And it's more nutritious and it's productive. So this is years and years of them taking a wild plant and domesticating it. Have you ever seen, how many of you have seen like a wild Okay, wild grapes we get around here, we get muscadine. They're certainly not like the grapes you get in the store. You get those nice little seedless grapes and the skin's thin and you pop them in your mouth and it's easy. Whereas muscadines, you got to work at them, right? The skin's really thick and the seeds are all in the middle. Have you ever seen any other kind of like wild vegetables growing and they're almost unrecognizable? Mhm.
I've seen wild strawberries.
Oh yeah, the little itty bitty things. Yeah, those are weird,
Wild
right?
onions.
Yeah, wild onions. Okay, yeah. Uh-huh. So they're just not what we're used to seeing because we've domesticated these organisms to make them more productive for us. So
And
here's another good example. Colored cauliflower. Anybody seen the colored cauliflower in the store? I've seen it. Really pretty. So why do we do this? This is hundreds of years of selective breeding. This one looks a little purple. This one looks a little orange, whatever. Let's breed those together and see what we get. Back to all these Mendelian principles, right? So higher levels of vitamins than white cauliflower, actually. That's another example. So here's the dogs. So look at dogs. We have 197 groups, 197 breeds, I'm sorry, that are recognized by just the AKC. So way more than that if you consider breeds that are not recognized by AKC, which is a lot of breeds. So we've actually categorized what the AKC recognizes into 7 groups. So who has a dog at home? Okay, what kind of dog do you have, James?
I don't know.
You don't know, is he a mutt? Just a mutt dog? Okay. Do you know what he's like he or she is made of mostly or?
We think some terrier.
Some terrier kind of things. Okay, so terrier is a whole group. Anybody know? A what kind? A Yorkie. Okay. Does anybody know of anything that goes in the terrier group? So Yorkshire Terrier, he mentioned. Know what else goes in there?
The little yippy dog.
Some of them are little yippy. Not all. What now? Jack Russells. Yep, that's a terrier. West Highland White is a terrier. American Bull Terrier, which is what we kind of know as the basis for pit bulls, actually. Terriers, go figure. All right, how about, who else has a dog? Anybody else have a dog? Jack, what do you have?
It's my grandma's and actually likes me.
Yeah.
But he's a mix between a poodle and I forget, is that a short dog that's real like
Oh,
fluff?
like a corgi or something, or?
No, he's it's so I think so, or it's 2 3-letter words. Like he's a poodle and then
Something, something, something.
I'll
Lhasa Apso or a
text my
Shih Tzu
grandma.
or something. Yeah, okay. So anyway, so poodles, we'll talk about poodles in a minute because poodles are a little different in that people like that they are hypoallergenic, but people don't necessarily like the look of them as much as maybe they're maybe not. Some people don't think they're as cute. The toy poodles maybe, yes, but the standards are kind of big and strange looking. So y'all know Ozzy. So Ozzy's little bitty thing. What are these 7 groups does he belong in?
Toy.
Toy, we guess. Hound is correct. What other dogs go in a hound group? Basset. Greyhound. Right. So what's the difference between a basset and a greyhound? Think about.
Larger.
Yep. So hounds are actually divided into, we consider hounds 2 varieties, sight and scent. So which would a greyhound be?
I think sense.
Sight. They're tall and they have great vision and they run really fast. Also whippets, Italian greyhounds, things like that. Borzois,
Bloodhound.
Afghans. Bloodhound is a scent hound. Scent hounds tend to have the long nose and the big floppy ears, right? Red bones is a scent hound as well, right? And then you have working, herding. Like what's a herding dog? Who knows a herding dog breed?
Shepherd.
Anything that says shepherd. Yeah, Old English sheepdog, German shepherd, Australian shepherd. How about a sporting? What's a sporting dog that you might know? Like a spaniel, things like that. This one here that says non-sporting, this is the catch-all. This is for the dogs that don't fit anywhere else. So
Where would golden retriever go?
that's a sporting.
Where would
Retrievers are
Samoyed
sporting.
go?
Where would what?
Samoyed.
They're working because they're meant for pulling. So that's the difference. So we have all these different dogs that came from a wolf, but they're all the same species, right? There's a ton of variety, but they're all the same species. Yep. Isn't that funny? So then we have the idea of hybrids. So what are hybrids? What's a zonkey?
I saw that like legit.
All right, so check this out. Your hybrids can be organisms with variety that are the same species or organisms with variety that are of different species. But if you're going to breed organisms of different species together, very often it's not going to work. It's only going to work if they have what? The same number of what? What do you think? Yes, chromosomes. It's only going to work that way. But typically this offspring is not going to be able to reproduce. So this is a zonkey, which would be a hybrid of what?
Zebra and donkey.
A zebra and a donkey. What?
I like my name for it better, Deborah, but that's, but they're really cool to look at.
So this is a zonkey. Look at his little feet. See his legs?
Hi, Deborah.
Hi, Deborah. My gracious. He's looking at her like, what? Leave me alone. I don't know what I am. I'm so confused. I don't fit in anywhere. It's cute though. So that's a zonkey. So a zonkey is obviously your zebra and your donkey. So the idea of hybrids was brought up by Luther Burbank way back late 1840s. Developed 800 varieties of plants. So hybridization is crossing dissimilar individuals to bring together the best of both organisms. Remember that hybrids are dissimilar, okay? And then you get individuals that are produced by crosses, and they're sometimes hardier than their parents. And then you can get this disease resistance and some of the things that he was seeing with his crosses. And then also you get a new line of plants that had traits that farmers needed to increase production. So again, this can be one species or two species, either way. Mules in particular, a lot hardier than horses in many ways, right? They use pack mules for pulling really heavy things, right? And that's the reason. So then here, let's look at this. So more hybridization. We got some cows. This here is a Brahma cow. He's from India, and they can withstand crazy amounts of heat, and they're chill with the heat, doesn't bother them. But this is an Angus cow. If you've ever had an Angus steak, you know that is some top quality beef there, man. That is good stuff. So we can throw them together and get a Brangus cow, which can withstand the heat and make good meat. So you can breed these and raise them in places that are really, really hot, which maybe wasn't a possibility before. So how about this? Two of the same species. What happens when you put a poodle and a golden retriever together? What do you get?
Labrador, he's super adorable.
Not a labradoodle, but a golden doodle, right? Known for being adorable, hypoallergenic. So this is why they breed them. One, they're cute. People like golden retrievers. Dog trainers have been known to call them lazy man's dog because they are just so willing to please. They are super willing to please. They'll do anything for you, but they also shed like mad. And if you have allergies, that's not great. Or if you just don't want to clean up behind them, that's not great. Poodles are one of the smartest breeds of dogs, strangely enough, that goofy looking thing. Now they have these poofs here because they're meant for water and cold weather work, and that actually keeps their joints warm. So that French poodle cut that you've probably seen and been like, what the heck? There's an actual utilitarian use for it, as strange as it looks. That's why they do it, keeps their joints warm. But they are hypoallergenic. They don't have fur, they have like a hair. So you get this little guy, right? Oh, so cute. This is not a breed. This is what we call a designer dog because he's a combination of breeds, but he is a type of hybrid. Now, what do you get here, guys?
Liger.
And what is special about a liger? I mean, obviously the golden doodle
Because it
was
sounds awesome.
cute, adorable. There we go. Thank you very much. You get a liger, and why? Because they're known for their magical skills. Tell me you know this line. Tell me you know this line, people. Known for their skills in magic. Who has not seen Napoleon Dynamite? So you can be ostracized. Okay, good. Everybody's seen it. Most of the first class, and you haven't seen it, have you? You just looked at each other like, oh, girls, you got to fix that. That's your Friday night job. You've seen it?
I've heard it's really great.
Amazing movie. You of all people would love. He hasn't seen Napoleon Dynamite. Who in this room would love Napoleon Dynamite more than Jack and Bonnie? I mean, come on. No, you got to watch that. All right, so inbreeding. So hybridization, we're taking dissimilar parents, putting them together. Inbreeding, we're taking similar parents and putting them together. This is all selective breeding. So why are we doing this? Because we're trying to take a characteristic and continue passing it, but it can create some problems. because it can bring together two recessive alleles. It can also bring together two dominant alleles sometimes that cause problems, which we're going to talk about in a minute. So you're doing it because you're trying to maintain characteristics of organisms. You don't want other stuff. So you're going to keep breeding what you like, but you're losing your variety that comes from your random assortment with your sexual reproduction. When you get your hands in it and you get all messy and you start choosing, right? You start engineering. So it ensures that these characteristics are preserved, but it can cause problems. So a lot of the members are genetically similar, and that's how you end up with two individuals that have two recessive alleles, or sometimes two dominant, which is what we're going to talk about here. Okay, so we're going to talk about dachshunds for a minute, because yeah, I love them, but also because they're a perfect example here. So dachshunds, not a species, a breed of a species, come in 3 different coats in like 5 or 6 patterns and probably 7 or 8 different colors. Most dogs do not have that much variety within their breed. Like if you've ever watched dog shows, Cocker Spaniels, they have like 3 colors, and you know, there are certain standards that breeds have to follow. Oh yeah, grab it. Also, I think you may have a binder in Lost and Found by the front door. Check on your way out, okay?
No.
All right, so that's okay, you're good. So we're going to talk about a particular pattern with dachshunds, because this is a really great example of inbreeding. So dapple is a marking on a dachshund that's similar to merle. If you've ever seen a merle Australian Shepherd, they're the ones with the patchy colors, right? It's spotty. It's really, really pretty. And the problem is, though, if you breed one dapple dachshund to another, you get what's called a double dapple. And it's really, really unique looking. It's very different. It's a double dapple if you breed one together. Yep. The problem is that the dapple gene is the merle gene. So they use an M when they categorize it, and it's a dominant gene. But a double dapple, 2 dominant M's, is bad. It causes all kinds of health problems, which we're going to see. So your you got your big M and your little m. So you had 1 dominant and 1 recessive. Your dominant is going to show that's your dapple. This is what dapple looks like. You can see he's a smooth. These 2 are long hair. So there's 2 of your coat types, right there. So this is what happens. It's going to be expressed, but this dog's cool because he doesn't have 2 dominant alleles. It's the dominant allele that causes problem. 2 recessive alleles, you're going to get a non-dapple dog. Now there's also another pattern in dachshunds that's completely different. It's called piebald. And that's like it gets involved in other things. It's kind of like a beagle where they have patches of color. But these guys here are either solid color or a particular pattern. So this guy would be what you call like a shaded red. See, he has shading on him, but he's pretty much a red. And this guy here is a wire hair. So that's the other coat, but that's called wild boar, this pattern. And it's sort of like a sable shading. And this is what Ozzy is, but he's a short hair version. That's the black and tan. So it's a solid color black, but the tan is called points. Okay. Now here's what you get with a double dapple. 2 dominant alleles for the dapple, and it produces a dog that looks like this. These are some examples. So you get 2 long hairs here and a smooth coat. Do you see this face is divided? It's got 1 blue eye and the face is divided. Most of the chest is white. Most of the chest is white. All the chest is white. Entire face is white. Most of the body is white. You see a dachshund where most of the body is white, or all of the face, or half of the face like this, or the chest, that's almost a dead giveaway. Now they do genetic testing, but that's like almost a dead giveaway. You don't want to breed that dog, and here's why. Because for some reason, when you put these 2 dominant genes together, what you get are health problems and abnormalities. Blind, or born with small eyes, born without eyes, born missing 1 eye, deaf in both ears, deaf in 1 ear. And then all of the typical dachshund health problems are all exacerbated. They're all worse. So things like intervertebral disc disease, brittle bones, Cushing syndrome, that has to do with how your body produces androgens, particular hormones, cortisones in your body. And then they can get thyroid issues and all these problems. So it's not a good practice to put them together. So intentionally breeding them is considered disreputable, and unintentionally breeding them is considered irresponsible. So how would you know if you had a dapple, so that if you had a dog that was carrying the dapple gene, that dominant M, how would you know what to breed it to? Because you could say, kind of as a standard, that you wouldn't breed any of these guys together, because you know they're carrying that dominant M. And usually you can breed to one of these guys and be good, but there is this really strange thing on occasion that's called hidden dapple. where it's a pattern kind of like this, and you just don't see it as well. It's not as obvious, but they still carry the gene. So what would you do if you were a breeder? How would you be sure that you weren't going to produce a dog with 2 dominant Merle alleles?
Genetic testing.
Genetic testing is exactly right. There are a couple places that do it. I think UC Berkeley is one. The biggest is called Embark. And you do it just like you do 23andMe or Ancestry.com. You swab your dog's mouth and you send it off, and they send it back, and they tell you exactly the genetic makeup of your dog, what genes they carry, especially for color and coat. So that's really good to know. So this is where a very practical application of genetic engineering comes into place. There are some people who intentionally breed dapple to dapple because they like the way the babies look, but they get, I tell you what, they get reamed by other people who are like, no, that is such a bad practice. All right, so how to increase variation? Well, you're going to increase variation by introducing mutations. So if you see a mutation in a population and you want it to continue, you're going to take that mutated organism and you're going to breed it, right? So you can increase the genetic variation of population by introducing mutations, which are the ultimate source of biological diversity. So if you had, let's say you bred cats and you had all black cats, and somebody had a white cat, and you know that that's a mutation from the typical gene, you're going to bring that white cat into your breeding program if you want to produce more variety, right? So biotechnology, application of technological process, invention or method. We've got all kinds of stuff we're going to talk about in a minute. So let's see, selective breeding, one form, important in agriculture and in medicine using radiation or chemicals. That's another way. So mutations are not always harmful. Just remember that. We all have mutations, right? Mutations are good things sometimes. So you may want a mutation. You may find a mutation that's advantageous. You may find a tomato that grows better in the heat, and you want that. And this is another example. Oil-eating bacteria. Have you seen this stuff? So you take a bacteria, and they've got bacteria that can actually munch up oil. So when there's an oil spill, it can get out there and eat it. So we use bacteria for lots of things because they're small. So you can grow them in a lab. If you can get them to produce a particular protein or perform a certain task for you, great, we're going to grow lots of them. So they can be treated with radiation and chemicals at the same time that can increase the chance of producing a useful mutant. So they do this on purpose. They zap them to try and see what they can get out of them. Sometimes they know what they're going to get out of them, so they zap them. Right? I mean, for real, right? So they produce hundreds of useful bacteria at this point. So some of them consume oil spills. You can take this, if there's an oil spill, you can toss this bacteria in the ocean and they will munch up the oil. Go figure, right? And then some of them are going to be used for other stuff, which we'll talk about in a minute. But working on bacteria that can clean up radioactive substances, this is something we're working on now. They can clean up radioactive substances and metal pollution in the environment. So like mercury poisoning, things like that. If we can find bacteria that can handle that, that's great. Oh, camera, you up? Poor Ethan is tired. All right, good. So we'll talk for a minute here about polyploid plants. So here's a word for you. Autoploidy and polyploidy. So autoploidy, small diploid seeded banana, contrasted with a large polyploid banana that produces no seeds. This thing at the top, this is a wild banana. This is what bananas look like before we got a hold of them. Now they look like this. The seeds are, pretty much non-existent. You don't really see them, right? I mean, they're in there, but they don't look like that. Would you eat that? Somebody ate that.
I just like bananas.
But you also got corn.
Yeah, I mean, look how big the seeds are. Isn't that weird? That's not what you're used to seeing, right? So that's the point before we got a hold of it. So other things we can do. Drugs that prevent separation of chromosomes during meiosis can be useful in plant breeding. So why is that? Because they produce cells that have many times the number of normal chromosomes, and then plants grow from these cells. So these are called polyploid because they have many sets of chromosomes. So polyploidy, isn't that a fun one? Polyploidy is usually fatal in animals. So we don't do it with animals, but plants can be better at tolerating extra sets of chromosomes. And they can quickly produce new species of plants, larger, stronger, faster, stronger than diploid relatives. So yeah, so this is just some things we've done with genetic engineering. We've done it with citrus fruits. We've done it with bananas. Think about, we've got watermelons with almost no seeds, grapes with almost no seeds. You know what we're doing, we think. So human manipulation of an organism's DNA. Here are some examples of ways we've done that. Recombinant DNA, we're going to talk more about that, cloning and transgenic organisms. All right, so recombinant DNA technology. So this is how this works. You actually take a plasmid DNA, which is this round thing here, and you're going to take DNA from two sources and join them together, and it changes the genetic makeup of whatever that thing is. So it's used with, we do it with restriction enzymes, and they're going to cut the DNA. So you have this plasmid DNA, it's a circle, okay? And you're going to cut it. Then you're going to stick in whatever chunk of DNA you want and stitch it back together. And then you can put that inside of a bacteria. And if the DNA you put in tells the bacteria to produce a certain protein, that's what it'll produce now. So now you can farm that bacteria to get the protein you want. So it introduces a new organism, new DNA to an existing organism's DNA. So this is a graphic of how it's actually
A
done.
common technique in genetic engineering is to insert a new gene into a loop of bacterial DNA called a plasmid. The molecular tool used to cut DNA is a restriction enzyme such as ECO-R1.
I love how these
The
look so
enzyme has a precise shape that allows it to
animated,
work
like
along the groove
animals,
of the
little
double
creatures.
helix. Scanning in the case of ECO-R1 for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted. When it cuts, ECO-R1 leaves a sticky end.
They actually call it a sticky
This
end. That's
helps
real.
the
That's a real
gene to
thing.
attach.
Sticky end.
All these complex
The joins are then stitched together by another enzyme called DNA ligase.
Settle down, y'all. Here,
The
stitch it
genetically
together.
engineered bacteria is grown in a culture medium. Very quickly, large numbers of the bacteria can be produced, each with a copy of the inserted gene. The bacteria duly manufacture whatever protein the gene codes for, and so the desired product is made.
So a great example of that is, who knows, anybody know someone who's diabetic, takes insulin? You know somebody who takes insulin? That's one of the main ways we produce insulin, right? Just like that. And we're going to do a little more of that in a minute. Transgenic organisms. Okay, this is when you take an organism.
Are we the ones that put the cutting enzyme in the?
Yeah. Yep, we do it. So transgenic organisms is a type of recombinant DNA, but it's when you take an, usually not a bacteria, but maybe an actual complex organism, multicellular organism, and you have it with a chunk of DNA that doesn't belong to it. So it possesses a useful gene from other organisms. So just like that cat at the beginning, this is a tobacco plant that has a gene from a firefly in it and makes it grow and glow in the dark, which is kind of weird. Like, why would you want that, right? There's reasons for it. We'll talk about more in a minute why you would want an organism to glow at all. And then we do have transgenic microorganisms, and this is specifically like we were talking about with insulin. Also growth hormone, clotting factor, things like this are grown in bacteria. We just stuck a little bit of DNA in there. Now the bacteria are producing what we want them to produce. So we get transgenic animals, we can get better meat, resistance to certain diseases. We can also use transgenic plants. And in this case, they can be really good for the food supply, and they're better for like certain climates, things like that. So 81% of our soybeans and 64% of our cotton are genetically modified. Soy is in almost everything. I mean, it's in almost everything, really, truly is. So, how did that override? I don't know. So genes for disease resistance produce their own insecticide. So there are plants now that we have genetically engineered to produce proteins that are essentially insecticides. So we don't have to spray those plants with pesticide and insecticide, which is dangerous for us to consume, right? Like if you don't want to go out there in a field that they just sprayed with something and, pick up that strawberry and munch on it, right? Because that's gross. But we now have plants that we can grow that no longer need the insecticide because they themselves produce their own shield. And then we can do things like weed resistant, heat resistant, increase vitamin content. Weed resistant is important too, because they resist weed killer, because you don't want weeds in your field of corn. So you have to spray for the weeds, but then that can kill your crop. So we have some now that you can spray for the weeds, but they don't hurt the crop. We've come up with all kinds of stuff. So here's another example. These little piggies here and these chickens. Yeah.
From anti-aging therapy in mice, animals carrying human cells, breeding the healthiest livestock, and building organs outside the body, genetic engineers are constantly searching for answers to many social challenges. From global issues to medicine, these guys put everything to the test. One of the biggest goals of genetic modification is to reduce world hunger by producing more food. These featherless chickens have been genetically engineered to keep them cool.
Since I'm a geneticist, I was looking for a genetic solution. That means instead of cooling the environment of chickens with feathers, is helping them feel cool by just removing the feathers.
Without feathers and scales on their feet, these birds can grow comfortably in hot countries like Nigeria and Indonesia. The hope is that once successfully bred, the healthy commercial-sized birds will help feed underdeveloped countries. To fight global warming, some turn to enviro-pigs. The industrial amount of waste from pigs is a serious environmental pollutant.
I really need it.
Pig manure has a very high level of phosphorus within it from normal diets to normal pigs. And that phosphorus usually is more than is required by crops produced on land fertilized with pig manure.
But the remarkable thing is that the enviro-pigs carry a man-made gene. One part comes from E. coli bacteria, which produces an enzyme that can break down phosphorus. And the other from mice produces a genetic switch that allows this special enzyme to be made in saliva. These two pieces of DNA were combined to form a new compound gene that makes their food more environmentally friendly.
Look what he said.
Medical researchers across the world are also turning toward transgenics for answers.
If you look at the word trans gene, it means that the gene was transferred, the foreign gene was transferred to the animal.
Having successfully used transgenics to insert a jellyfish gene into a rabbit, when charged
Very
with
glowing
glucose
animals.
violet light, the skin and the eyes of these rabbits glow green. But why make animals that glow in the dark? The incandescent gene allows researchers to easily track the movement of cells. By tagging certain cells with the glowing jellyfish genes, scientists can track which cells go where after an organ transplant. The technology is helping to find treatment for problems like blindness and bone disease. Glow-in-the-dark rabbits are just a start to the medical challenge that genetic engineers have taken on. No one said it was easy being green.
So obviously, when we start talking about this kind of stuff, we come up with a lot of ethical questions, right? So when you hear they made a cat glow or they made a rabbit glow, it sounds just like unnecessary. Like, why would you do such a thing? But there's a reason, right? When you get organ transplants, it would be really helpful to know exactly where those cells grow when an organ that's been donated connects into an existing body. How does that work? Where do all the parts move? Because obviously they're going to grow into one another. And knowing exactly how that process works could probably help prevent a lot of things like rejection and maybe help people get better fit. I'm sure there's all kinds of stuff that they can learn. I don't even know enough about organ transplant to know what you could learn, but I'm sure there's endless stuff that you could learn by seeing exactly how they grow. So that's one way to do it. You tag those cells glow in the dark, and now you can see. So cloning has a lot of ethical stuff, and we'll talk about this too, but it also has some really, really interesting potential benefits that we're getting closer and closer to. So this is Dolly right here, and she was the first cloned mammal. And this one's close to my heart because she was cloned in 1996. So I was right smack in the middle of my biology degree when this happened. So you can bet this became one heck of a conversation in my classes. Like it was the news, and it was all we talked about for like, and we got into all the ethical discussions and how was this done specifically down to the microscopic level? How did they do this? And why would we do this? So I really have always enjoyed the topic of Dolly in particular. Yeah. So a clone is a genetically identical organism. So technically an identical twin is a clone, but they're developed when you have one that divides into two. That's not how Dolly was made. The first clones were amphibians, tadpoles. And obviously this makes sense because this is done outside the body, right? They're fertilized outside the body. They grow outside the body. None of it's internal with amphibians. So you can really work with them in a lab, in a dish, and see what's going on. And that was all the way back in 1952. So that's been a while. But it was the Scottish scientists who managed to clone Dolly. And this was in 96. So she did suffer from lung cancer and arthritis, didn't live, wasn't super healthy, and they did have to put her down in 2003. So this is something they've seen in other cloned mammals, is a tendency for health problems. So since then, though, they've managed to clone other mammals like cows and pigs and cats and mice and all kinds of fun things like that. Now, here's how it was done. So what you do if you want to go ahead and clone you a sheep is you, in this case, what they did is they got themselves an egg, okay? And they emptied the egg out. They basically went in and sucked out all the genetic material that was in the egg. And of course, you do this under a microscope with an itty little needle. Sometimes it's one of those glass pipettes that you've strung out to the point where it's almost like a hair, very, very small. So they go in and they empty it out. And then the next thing they do is they obtain a somatic cell from the mother. So an autosome or a somatic cell, a body cell. In this case, the cell they picked from the mother was an udder cell. Yep, her udder.
Okay.
Don't know why they picked an udder, but they picked an udder. So then they take the somatic cell, take the basically all of the guts out of it, and they stick it into this now empty egg. All right, and then they take that empty egg and, well, they let it grow into an embryo first, and then they take the embryo and they implanted it in a surrogate. What's a surrogate? Anybody know what a surrogate is? Like a middleman. That's an interesting way to put it. Middleman for a pregnancy. Okay. Think about that one for a minute. That colloquialism doesn't quite work there. Yeah, but yes. So a surrogate is.
It's a to use more nondescript language. It's like using a different oven.
Okay, using a different oven for your baking. Okay, okay, there's an. So
baking
basically, a
ingredients in there.
surrogate is a female who carries an offspring that doesn't genetically belong to her. So there was a surrogate sheep in which this cell was implanted. So are there human surrogates? Do we have human surrogates? We do. We do have human surrogates. So there are some women who, for whatever reason, cannot carry a pregnancy or don't want to carry a pregnancy or whatever the reason is, and you can have a family member be a surrogate. Sometimes a family member or a friend will do it for you, but you can actually hire a surrogate. You can pay a surrogate and they make good money because they use entirely medical processes and they implant an embryo, and some other person who's your surrogate will carry your offspring to term and deliver for you. And I actually knew a lady, there was a kid at the gym whose mom was a surrogate. And when he was on the team, she was on her third baby that she had grown for someone else. Kind of interesting. Wow, that was wild. Yeah. So you end up with genetic identity that is equal to that of your somatic cell donor, which the question is now, which one of these is the mother, the surrogate that carried it, the genetic donor? Technically, it's the genetic donor in this case, would be the mother.
They have to go into a TV show.
Mhm.
They have to yell and argue about whose kid it is for
Yeah.
like at least 50 minutes of
Yeah.
the hour-long TV
Jerry
show.
Springer style. Throw a chair
Yeah.
or two. Have to throw a chair or two.
Such a fun
The bouncer has to come in because
And
it's
then
only good
that-
if he shows up.
Calm down, talk it out,
Yeah.
and then realize it's neither.
And then Maury Povich needs to come out with yeah. Okay. But there's no paternity.
Mhm.
Who is Dolly's father? Technically, Dolly's father is the father of the donor father of the donor sheep. So when you think about it,
It's the Dalai Lama.
it's the Dalai Lama. There's that too. So what are the issues with cloning? All right, so animal clones tend to show issues: high death rates, deformity, disability. Dolly was the only success out of 276 tries in this lab with these scientists.
Did they
That's
just
a
choose a
big
different
number.
body part to
So
get
that
the
means,
cell
yeah,
clone each
so
time, and that's how they landed on
some of
target?
it could have been different, different somatic cells that didn't work. I don't know how they're categorizing 276. Is it start from the attempt to collect the somatic cell genetic material? Does it start from the attempt to remove the nucleus from an egg, or does it start from the embryo? Perhaps there were 276 embryos that didn't survive to be implanted. So is cloning a natural process? What would you all say? No, unless it's like the creation of an identical twin. It's really not. or a offspring of something that produces asexually, but certainly sexual reproduction, cloning is not natural. So would you want to be a clone? Would you want to be the genetic clone of another?
I
Uh-huh.
don't like lung cancer.
Huh?
I don't like lung cancer.
You don't like lung cancer?
So, yeah,
Okay,
yeah,
so probably not.
no, that's not my favorite thing. to have.
Yep, yes.
If we got rid of the death, poverty, and disability, then you.
Then maybe.
We can talk about it.
Then we can talk.
Yeah, what were you gonna
But
say?
until we get rid of the chance for us to die.
Yeah, that's not great.
horribly,
Well, you're gonna die anyway. I tell you that, but you're gonna die.
Yeah.
We're all gonna die.
Spoiler alert, Alex.
Yeah. Hate to break it to you, Alex, but yes, Jack.
If Hollywood's taught us anything,
Ah,
look at Star Wars.
yes, this is true. Watch your feet.
The clones
She's coming back.
did not half bad, but really, probably not because,
So this is about being a clone. How about, would you want to have a clone?
No,
Stunt double, body double.
No. A body double?
Oh, that's a good
As far
point,
as
though.
So,
That's a good point, though.
I mean,
So-
the
hang on,
key
Cole's
word
answering. Uh-huh.
Key word here is do you want, and you don't really get that want.
You don't get the one if you're somebody's clone, correct? If you make a clone, you have a clone, you may have a choice, but certainly if you're a clone, you don't have a choice. So here's a question. So here's where a lot of the ethics come in. So there have been cloned human embryos that are used for research. Is it okay to just clone a bunch of human embryos and then destroy them when you're done? That's an ethical question. These are the things that come up when you start getting into this type of technology, because you're creating life. And obviously they do freeze embryos. There are lots of human embryos right now that are cryogenically frozen, and they're like literally frozen in frozen animation. Like, I'm developing. Nope, I'm not going to develop. And there are people that will do this for various reasons. There are people that will do, fertility work with doctors, and they will get viable embryos, which then you get the ethical question. So have y'all heard of Octomom? She was before your time, but have you heard of her? So right at the point when like in vitro and all that type of fertility doctor help came to get pretty reliable, they got to a point where doctors had to start making really hard decisions, because they would get a whole bunch of embryos. Okay. They'd have 12 viable embryos. Well, if you implant all 12 viable embryos into this woman, chances are they're not all going to survive. Well, how many are? The better the technology got, the more of them survived. So there was a woman in I think it was late 90s, she may have been early 2000s, but she had 8 viable embryos that all survived, and she delivered all 8 babies. And it was crazy, because she became like a TV sensation, and she went on all the talk shows, and then she got even weirder, and she had like the big collagen lips, and I don't know, she got really weird. But anyway, who wouldn't if you had 8 babies at one time? That's a litter. That's not what human beings are built to do, right? That's necessarily not normal. So then you get to the point is, well, do you freeze these embryos that you're not going to use? Do you destroy these embryos that you're not going to use? Do you freeze them for your use later? Do you freeze them to donate them to someone else to use later? It's your genetic material. So all these questions come up when you're working with human embryos. And when you start cloning, now you're talking about human embryos that have the DNA of an existing human being. So it adds in a whole other level of complication. Yes.
Well, going back to like the surrogates, like the ladies get paid.
Yeah.
And going back to the clones, it
Mhm.
would also depend on like if I was.
Are you getting paid? How much you getting paid?
Putting like
Mm-hmm.
Well, I guess if I would be a clone,
Yeah.
then I wouldn't need to get paid, because I would probably.
So here's where cloning yourself may become more appealing. So what if you can clone your own cells and produce organs and tissues to replace damaged organs and tissues? That's a little different now, because now you're talking about your own tissue that you're putting back into your body. Say you were in a terrible car accident and 50% of your body was burned. Okay, so you can get skin grafts from your own body, where they can take chunks of your skin from other places and move it around. You can get grafts from donors, like cadavers, things like that. They have some stuff now that's like a man-made stuff that's similar. Or what if you could just grow your own skin in a lab, and then just have your own skin slapped back on you? That may not be such a bad idea. So has anybody heard of cord blood banking? Do you know cord blood banking? So umbilical cord blood does contain stem cells. So one thing that people started doing probably 20-ish years ago, and they're starting to do there was a really big bang where it was like the new thing and a lot of people did it. I think they're probably doing a little less now because it's not as hot a topic, but they'll draw some blood from an umbilical cord and it can contain stem cells, and then they'll just go cryo freeze it and it just stays. And it just stays there. Most people's is just staying there. Because the idea is that as science continues to advance, there may be a use for that in the future. Maybe you can use it yourself, maybe a sibling or a child can use your stem cells for something like recreating an organ that's been damaged. And I think certainly in your lifetime, maybe not by the time you're having kids, maybe though, it'll be to the point where the technology is getting so good, it'll be to the point where this type of thing becomes more and more commonplace, where people will bank something like cord blood or some other type of stem cell or genetic material for the purpose of replacing their own body parts. Because we can 3D print organs now, and that's pretty awesome. And we're starting to do this stuff more and more and more. So if you have something goes wrong, you have some physical problem or whatever, then you can replace it with your own stuff. All right, now I got two asleep. Yo, Alex, Ethan. All right, yes, Jack.
So I lost my whole train of thought because he said 3D print organs. That made me think about the 3D printed chicken.
Oh,
But
there's that.
Yeah, I think
I should stop saying that.
possibly. But I feel like if you could clone your own cells, that would be handy, especially as the science gets more and more developed.
I think it's going to get pretty good. I think probably we've seen huge advancements in technology in the last just 100 years. I think in my lifetime, ridiculous advancements in technology, just in general. This type of thing, I expect to really become, I expect this is going to become more commonplace. It may be the kind of thing that when you're elderly and you're dealing with, typical problems like bone loss or whatever, they'll just say, oh, well, here's some of your own bone cells. And now you don't feel so bad. You don't hurt everywhere. Like my dad had bone loss in his jaw and he had dental implants and they didn't stay. He pulled his own teeth out of his mouth because they were falling out, and he's, these days, but his own teeth came out of his jaw because he didn't have any more bone in there. That's the kind of thing that we use bone grafts now for this kind of thing. They have a powder that's like a grafting powder that they put in there and it helps grow bone. What if it's your own bone? That's even better, right? For you at least. So benefits of cloning, like we were saying, it can also reproduce animals with specific benefits, especially if they're rarer. And you can mass produce those animals, like those chickens with no feathers that can grow in countries that are underdeveloped or have a lack of food. Would be great for them to be able to have chickens that don't die because of heat over there. They can't afford to throw in air conditioning if they can't afford to get chickens to farm. I mean, gotta think about it. What's going to work best? And repopulating endangered species. That's a really good point too. Obviously, you got to be careful. You don't want to throw a bunch of clones in one place. You're going to lose your genetic variety in that species, which may be why they're endangered anyway. Who knows? But still, you want to. That's a possibility. It's something to consider. And then produce things like whole human organs from a couple of cells. Fascinating idea. Stem cell research using cloned embryos. Again, you have to think about that. Is this what we want to do? So here is some more, here's some other ethical issues. So possible outcome of Human Genome Project, no more genetically imperfect people like Stephen Hawking. Well, no more depressed people like Vincent van Gogh. Well, no more physically challenged people like FDR. Well, no more blind people like Stevie Wonder. Well, no more people with multiple handicaps like Helen Keller. Well, maybe there's a lesson here. So something to consider is, just because you can fix all the problems, does that mean you should? Which things are really problems? How do you define it as a problem? And, we certainly would never say that people that have certain disabilities don't contribute, because they absolutely do contribute to society and humanity as a whole. At the same time, there are some people who live lives that are just full of suffering. If you can prevent that, would you want to prevent that? All ethical questions, and they get really sticky, really sticky. Bioethics is a whole field, right? So here's some practical applications of genetic engineering. So how can we benefit agriculture? How can we benefit human health? And of course, identifying, we know this one, So agriculture, 93% of the corn you eat is a GMO. We're going to talk about GMOs for a minute. Oh no, hotly debated contested topic, GMOs. Oh, they're going to kill us all. No. So we've had 20 years of research on GMOs. We don't see any reason to believe that they're toxic or that they cause problems with fertility or reproduction. And they allow certain benefits, like we've talked about, crops that are resistant to diseases, crops that are resistant to pests, herbicides, crops that don't need insecticides. So these are all benefits, right? So everything we eat and much of what we wear has come from living organisms. So we can use genetic engineering to try to improve products from plants and animals. And ideally, genetic modification could lead to better, less expensive, more nutritious food, as well as less harmful manufacturing processes. So stuff like we're talking about with the, corn that doesn't need a pesticide on it. And then also things like, pigs with less phosphorus containing poo, or, oh my goodness, featherless chickens that look really atrocious. Well, it would be great if you lived somewhere where it was super hot and you didn't have any chicken and you were hungry. I would take a featherless chicken. Hungry is hungry.
Before you eat it, you make it.
Well, I guess it's true. It's going to get that way anyway. So genetically modified crops were introduced in 1996. 1996 was a big year for genetics. 2007, genetically modified crops. The stats in that year were 92% of your soybeans, which are in almost everything, by the way, 86% of your cotton and 80% of your corn grown in the US. So we use bacterial genes to produce certain things like this BT toxin, and this is the one that's harmless to humans, but has an enzyme in insects that converts it to a form that kills the insects. So this is an example of this is a plant that doesn't need an insecticide because it produces its own. If you try to eat it, it kills you. Hey, that's pretty good if you're an insect, but not if you're a human. So no pesticides are needed. You can produce higher yields of crops. And then there are ones that are resistant to herbicides, resistant to viral infections, produce foods that are resistant to rot and spoilage, and you can produce plastics for manufacturing industry from plants. You see the bioplastics, like the biodegradable water bottles and biodegradable plastics, those types of things. That's how we get that. So your cows, 30% of your cows out there today are injected with hormones with recombinant DNA that make them produce more milk. And that can be good for the cow because that way it doesn't get, worn out and milked to death and whatever. But you're like, I don't know. Do I want milk from a GMO cow? You got to consider. Pigs produce more lean meat, high levels of healthy omega-3s, so we can make the pig meat more nutritious. Pork. Salmon, salmon. We've given salmon the ability to produce greater growth hormone. They grow more quickly and they grow in captivity. So we're not out catching wild salmon, although you can get wild salmon. By the way, wild salmon and salmon grown in captivity are different colors. Next time you're in a grocery store that has a good seafood section, go take a look. Compare your wild salmon to your farm-raised salmon. Your farm-raised salmon is much paler color. The wild salmon tends to be a very bright orange color. Yep. So spider genes can be put into goats to produce silk from their milk.
I read this.
And we're going to watch a really cool video about that because it's one of my favorites. It's a neat one. And then we can also put human genes into goat milk and we can get antibacterial goat milk. And in the future, we're hoping to clone more transgenic animals to increase the food supply, save endangered species. And in case you didn't know, in 2008, the US government approved sale of meat and milk from cloned animals. So most of what you've eaten probably. So here is this fascinating little tale, which I really love.
Not Spider-Man,
Spider goat.
You play goats.
Spider
Oh,
goat.
come here.
Spider goat.
Play the video.
Spider goat.
You cute little goat.
So is that Spider goat?
It is.
I'm Danielle Gatzweig. I am a master's student at Utah State University, and I work in Dr. Randy Lewis's spider silk lab. We are trying to produce the proteins that spiders use to spin all of their fibers. This is the spider that we base all of our genetic sequences off of. It's called the Nephilah clavipes. It's also called the banana spider. You can't farm spiders, they're cannibalistic, and you cannot get actual protein from them. So what we're trying to do is produce all those types of silks in other hosts. Those hosts include alfalfa, bacteria, we use E. coli, goats, and silkworms. The goats are not affected adversely. They are genetically modified from the time of conception, basically. So they're modified in the embryo, And then everything after that just comes from breeding. So when we breed the goats, not every baby has the gene. The only difference in the goats is they produce a lot of proteins in their milk. These goats are special because they produce one extra protein basically in their milk. So yeah, you tasted it, you look at it, you look at them, they look exactly the same. Spider silk is biocompatible, which is one of its really cool properties. So potentially we could use these products for medical uses and replace things that are used now, make them better, make them stronger, make them easier on your body. We've also had interest in the textile industry and in adhesives, making eco-friendly adhesives that don't have harmful byproducts, that aren't adhesives that aren't petroleum based. That's kind of the great thing about this research, is the kind of opportunities are endless, because it has the potential to do so much good. It would be a shame to not harness the power of genetics.
There's that word, power.
Interesting, right? You think about spider silk tensile strength wise is like, it's stronger than steel cables. It's really, really strong, especially those flipping Joro spiders. That silk is crazy strong, right? I mean, crazy. You've tried to break down any of those webs that came around this summer, that gold stuff.
It was like
Oh,
hair.
yeah.
At my grandma's house, it was a yellow.
Yeah, it's golden. Yeah.
And it went from the tree all the way down to the ground.
Yep. It's pretty crazy. Pretty crazy.
The power lines when it's foggy or when the sun.
Yeah, and they're all there. So basically they can milk the goats, and then they can isolate the protein from the milk and pull, basically string it together and produce the spider silk from it. Really neat idea. Like right now we have sutures and stuff that we use, internal sutures. If you've had a surgery, they're internal sutures that sort of, they sort of dissolve into your body.
They use that for your mouth too.
Yeah, they do. Yeah. But imagine like having sutures made of something like that. Imagine having sutures that strong.
Like the wisdom teeth ones, they dissolve.
Yeah, they dissolved, right? So here's some other uses for medicine. So early physicians extracted substances from plants and animals to cure patients, right? 20th century, we had vaccinations coming in, saving countless lives. So today we have recombinant DNA technology sources, which is the source of some of the most important advances in prevention and treatment of disease. So our goals here are to prevent health problems. Infant blindness was one in particular that was mentioned when I was looking through this stuff. So transgenic plants and animals can be used to make human antibodies. In the future, transgenic animals may supply us with human proteins that can be used in disease prevention. They can simulate human diseases caused by defective genes. Now, this is important because if you can simulate a disease in a lab animal, that's how you go about figuring out whether or not your medications or your treatments are working. So we will produce animals in a lab that are models. They're model animals, mice or rats or whatever, that are diabetic rats, and whatever, they have whatever disease you're trying to work with. You can produce that disease in a lab animal so that you can see whether or not the treatment you're trying to work on actually does what you expect it to do. So we use models based on simulations to follow onset and progression of diseases too, and to test new drugs like Alzheimer's and arthritis. These are things we've worked with a lot. And then recombinant DNA technology can make important proteins that could prolong or save human lives. Human growth hormone is a good example. Used to treat pituitary dwarfism, which is not the same thing as achondroplasia. This is when your pituitary gland just doesn't function and you don't grow. And it's now widely available because it's produced in bacteria, just like insulin. Same thing we talked about, right? And then blood clotting factors. Say you were a hemophiliac like we had in our lab last week. This is the kind of thing now that you would be treated with. So this is about biologic medicines. And it's how it's made, so it's good. I could ever watch these like all day long? Oh yeah.
Medicines are proteins produced inside living cells, which are mini factories generating the genetically engineered protein treatments. It's a different approach from traditional drugs, which are manufactured using chemical synthesis. Biologics offer hope where other therapies have failed. To mass produce these biologic medicines, scientists use the molecular machinery inside living cells. The process starts with the insertion of genetic material into Chinese hamster ovarian cells. They're known as CHO cells. They keep them frozen in liquid nitrogen until the next step. In the seed lab, a technician thaws the cells in warm water. He transfers them to a glass flask that contains growth media. The growth media is comprised of hundreds of nutrients, including vitamins and minerals. It's a recipe to help the cells multiply. A worker transfers the flask that contains the cells to the production side of the plant. The team injects the mixture into a 5-gallon capacity bioreactor. Inside, a propeller agitates the mixture to stimulate growth. Over a 3-week period, the number of cells increases exponentially. Each new cell is programmed to produce the target biologic protein. Next, a technician measures salts and other dry ingredients. He's preparing a recipe for purifying the protein liquid once it's been separated from the cells. He labels the bag and records the information separately, as required by government regulations.
Important to say that.
He adds the ingredients to purified water in a mixing tank. An automated system keeps tabs on the condition of the purification solution. After separating cellular material from the protein mixture, the team pumps it through steel and glass columns. Here, the purification solution works with resin beads to separate the protein from the impurities. Finally, this technician takes a sample and conducts a test to ensure the medicine is sterile. Grown in living cells, these biologic medicines are now on their way to potentially making a difference in people's lives.
So biologics are relatively new. And a lot of them, as you'll see here in just a second, have an ending that's like izumab. You'll see that in a lot of the names, or umab in some way. So here are some examples of, these are the most common biologics that we use here in the US. Any of these look familiar to you? Do you know any of them? Do you know what any of them are for?
I've seen Humira commercials and Elasta and Avesta.
Mhm.
Lucentis and
Anybody know what Humira is for?
I'm trying to remember what the commercial had.
Autoimmune stuff, but namely psoriasis.
And then I remember Lucentis.
Enbrel's the same. How about Herceptin?
Is that for?
It is. Yeah, you can guess from the little archer.
I've heard the first second.
I think this one is used for, I think it's used for cervical, something with cervical cancer or something along those lines. I think Avastin is a breast cancer one they use for treatment in there, but these are all different. So this is obviously for diabetics because it's a type of insulin one. So yeah, these are all different types of biologic medicines. And these are they're much newer for sure. They're definitely a newer class of drug. They've probably are making their biggest strides right about now. They're coming out with new ones constantly. You've probably seen the commercials where it lists like 9 million side effects, like for every drug, and you're going, oh my gosh, right? But these can be absolutely life-changing for people. So I've got one for lupus stuff that has totally made my life like 90% better than it was. Nothing's gonna make it perfect, but boy, is it better. The other thing is, you saw how much went into that, right? Making one of these. These are not cheap at all. The R&D for these things is crazy, right? You put in all this research and development, it's a lot of work. So yeah, these are pricey. So for some people, they're cost prohibitive. Thank goodness for insurance. But so mine is a once a month injection without insurance. Does anybody want to guess what the once a month cost without insurance would be?
5,000.
It's about 6,500, yeah, per month. So some of them are higher like that. Some of them are in the tens of thousands. So yeah, they can be really, really, really pricey, but they can also be a total game changer for people. So gene therapy. So this is the process of changing a gene to treat a medical disease or disorder. This is nowhere near as developed. So absent or faulty gene is replaced by a normal working gene. allows the body to make a protein or enzyme that's needed, and eliminates the cause of the disorder. So an engineered virus that cannot reproduce or cause harmful effects is what's used. You put the DNA with the therapeutic gene into the modified virus, you put the virus into the patient's cells, it inserts its healthy gene into the targeted cell and corrects the defect, delivers the gene that works correctly over a long term is the idea. High risk, experimental. We aren't quite there at being proficient at this yet. So it's not quite the accepted treatment that we're doing all the time now, but it's something they're looking at. So genetic testing. We can use specific DNA sequences to detect complementary base sequences found in disease-causing alleles. We can also do tests to search for changes in cutting sites for restriction enzymes. These are all just some things we can do. Some tests are used to detect differences between the length of normal and abnormal alleles. So these are some ways we can test. And we do have genetic testing available for diagnosing hundreds of disorders. So we can now diagnose hundreds of disorders with genetic testing, which is pretty cool. Now we have to talk about active genes. You've got two alleles, right? And for your mono genes, right? You have two alleles working and they're not both going to be turned on at the same time. Kind of like how we talked about active and inactive genes when we talked about stuff like, remember we talked about the tortoiseshell cat, one's turned off, one's turned on, that kind of thing. Well, maybe the one that's turned on is causing problems and you want to turn it off. So how do we do that? So we have ways to do that too. We can take that identical genetic material. Some genes are not active in every cell, and then we can take what they call DNA microarray technology, and we can use it to determine which ones are on, which ones are off, and try to downregulate or upregulate the expression of a gene based on what outcome we want. And then, of course, we can use it for personal identification. This is the lab we did, right? Can totally do this. Complexity of the genome, no individual is exactly like any other. So we use specific genes, right? The ones that tend to have more variety. We don't use just like any old gene. And then this is DNA fingerprinting. So this is what we did a couple weeks back, analyzed certain sections, looks to find out, whether you're a match or not a match. And you can get these samples from blood, sperm, tissue, or hair if it has a root, only if it has a root. So if somebody attacks you, scratch them and pull their hair out. There you go. That way you'll have everything you need, hopefully. Yeah, more than need. So this is the first case that used DNA fingerprinting.
November 1983.
Good voice for
The
this.
body of a 15-year-old girl, Linda Mann, is found in a village near Leicester.
Oops.
It's still war. And I can't believe that after 5 years. She's such a lovely girl. It was wrong.
3 years later, and a second girl is found less than a mile away. Dawn Ashworth was also 15.
When Dawn's body was found, police knew straight away they were looking for a serial killer. What happened during the course of this investigation would change police detective work around the world forever.
David, just to explain what was part of stuff here, these are mementos from the case.
People are screaming at me.
David Baker, who's now 84, was leading the double murder investigation. He'd arrested a 17-year-old, Richard Buckland. Buckland had admitted the second murder, but there was a problem.
There was no circumstantial evidence against him on the first murder, and he totally denied it.
You were convinced it was one murderer?
Yes.
David had read in the local paper about research being done at the University of Leicester. Professor Alec Jeffreys had attached DNA from cells to photographic film. The result, each person's individual DNA could be read. David Baker contacted Professor Jeffreys to get Buckland's DNA tested.
Nobody had ever attempted a crime scene DNA analysis, a real-life crime scene DNA analysis before.
Would the test find the killer? Would Buckland's DNA match that found at the crime scene?
They came back not connected. The result was stunning.
A blow to you?
Well, it was a blow because it meant that we got the wrong man.
Buckland was released. The very first time DNA profiling was used in a criminal case, it exonerated the suspect. David Baker was back to square one. 2 murders, 1 killer, no suspect.
It says, will the killer strike again? How can you help to track him?
We made a decision there and then to use the DNA system as part and parcel of the inquiry.
What happened next was extraordinary. Police asked all the men who lived in the local area to give a sample of their DNA. More than 5,000 men gave a blood sample voluntarily, and eventually the killer was caught.
This is Pitchfork. He went from being a very normal family man to a murderer in split second.
Colin Pitchfork, a local baker, was sentenced to life, the first person ever to be convicted on the basis of genetic evidence.
Today, there are ongoing debates about DNA, how the information is stored, who should have access to it, how it's used. But Linda's family have no doubts about genetic profiling and the historic role it played in solving a double murder.
So you guys know about the Innocence Project? Do you know about that one? So the first time DNA was used in a criminal case, it was used to exonerate somebody. Innocence Project goes to people who are already in jail, who have been accused of something that they still swear they didn't do. And they go back and take DNA from the crime scene that may have been in the 70s, 80s, or 90s before DNA was typically used. And they will run that DNA to see whether or not that person actually did commit that crime. They've exonerated, I think it's up to dozens by now. Some of them were death row people, like people who were set to be put to death and did not do. Clearly, 100% did not commit whatever crime they were convicted of having committed.
I'm big into true crime. I watched Forensic Files at like right when I woke up.
I got
at
some
6
podcasts
a.m.
for you, man.
And some of them still say they didn't do it, and then I watched some who are like death row stories that are like people who say they didn't do it and they have gotten it, and they've gotten sufficient evidence enough to go to trial and still get like turned down. It's really like at some points it's like that's unfortunate, but at the same time it's like these families.
It's a big deal. And when DNA first became a thing in the legal system, like, okay, if you were 18 and you were thrown into a jury box today and somebody said to you, this man's DNA was found at the crime scene, you'd go, okay, I did it, because you have a good enough understanding of it. But when they first started doing this in the 90s, people were like, okay, and they're all, okay, this is what DNA is, and this is how it's only for the, well, how can it only be for this guy? We're all human, we all have the same. Well, no. They had to explain it like at a kindergarten level every time for years, because people had no idea what it was or how it, how it could be used or what it meant. So it was a really big challenge for lawyers in the day.
Get real touchy when you condescend to them.
Yeah.
So sometimes they might have said it, they're like, well, that makes sense, but the way you're talking to me.
Yeah. So we've done fingerprinting since the 1980s. It's precise, it's reliable, it's used in forensics, obviously. It's used to solve crimes, convict people, overturn people. And then also we've used it, for example, there was hunting and we're trying to figure out what's going on with the hunting. So which herds did the black market ivory come from? Which elephant herd did this come from? Where do we need to go watch? There's ivory found on the black market. They're not supposed to be hunting these elephants. How do we know where they got it? We can find out exactly which elephant herd he got it from. So now we know how to look for the guy or girl or whoever, because we know where this person was. So selective breeding works because of the natural genetic variation in a population. Is that one going to be true or false? Oh, you've got your answers. Yeah, that's a true one. Hybridization crosses similar individuals to bring together the best of both.
False.
It's dissimilar, right? Individuals produced by crossing dissimilar parents are purebreds. No, those are going to be our hybrids, right? Continued crossing of individuals with similar characteristics is hybridization.
Inbreeding.
Inbreeding, that's right. And inbreeding increases the risk of genetic defects. That's true, it does. So selective breeding, we have our two main types here. We have hybridization, which is crossing dissimilar. And then what are some examples? These are some examples from your book. They talked about Burbank potato. I don't know. And then inbreeding would be when you're continuing to breed with similar characteristics, and your book had an example of Alberta peaches. So for biotechnology, get this out of the way. So this is a concept map. If you had done this one, this one's from your notes. So it's the application of technological processes, invention, or method to a living organism. So which can increase genetic variation through what? It can increase through introducing mutations or by preventing chromosomes from separating during meiosis. And then when you introduce mutations, an example would be things like this: radiation for your, zapping the bacteria we talked about earlier. Also, in this case, drugs that induce polyploidy, or which can yield stronger, larger plants. So genetic engineers can transfer what from one organism to another? In this case, it was genes, right? As a first step towards finding a gene, Douglas Prasher studied the amino acid sequence of a protein. Pressure found that the mRNA-based sequence that could have been protein. Okay, I don't know which one of these I did next. I think it was this one. We'll see. Yep, there we go. So Argobacterium can cause tumors in plants. So we've used recombinant DNA and transformed bacteria, which inserts itself into the DNA of the host cell chromosome, and then you get a completely new plant, or a complete new plant. So here's our little dolly again, right? Here's our little dolly. So the egg cell is fused with a cell taken from another adult or somatic cell, or autosome, you could say. The nucleus is removed, the cell begins to divide, and then you get this embryo, and then they stick the embryo inside another sheep, and boom, you have Dolly.
Does that mean that you could say that Dolly had 3 mothers?
Technically, genetically, she has one mother, but then she also had a surrogate mother. And the
Well,
third mother, her grandmother, are you saying?
No, the
if
other
she's
egg.
cloned from
Oh, the
one
other egg.
sheep,
Yeah.
then the cloned sheep mother would also be her mother.
So there's
Yes, that her genetic mother is the sheep from which the udder cell was taken. That would be her genetic mother.
And there would also be the mother
From the egg donor mother, but they took all the genetic material out of that egg. So that would be somebody who donated an egg cell, but wasn't a genetic donor because the genetic material had been, see, doesn't it get fun?
But there's also, and then there's also the mother of the udder cell donator.
Yes, and
Also
there's,
a mother.
yes, and
So
yeah,
Dolly has lots of.
exactly. It gets interesting.
all of them go on stage and start fighting and cheering.
Yes.
So 2 examples of how genetically modified organisms can lead to more environmentally friendly agricultural processes. Don't need pesticide and things like leaner meat in pigs. So should you be able to keep your genetic information confidential? State 2 answers, one giving a yes and one giving a no. A yes is pretty easy. I think most of you guys would be like, yeah, it's mine. So what do we come up with for no?
Because it can help with crime scenes.
Crime scenes?
And also you have like 2nd, 3rd, 4th cousins that you don't even know.
Okay, there's that. Anybody else have any other no's?
Your DNA can help with others through scientific research, so you should be forced to give it to whoever can use it.
Okay, there's that. Okay, yeah. So if the police want your DNA, they have to have a warrant. You know that, right? Like it is considered private to the point where they have to have a warrant to get it. A couple of years ago, there was a video that went viral of a nurse that refused to draw blood. Did anybody see this? There was an unconscious man that she was treating in the ER, and a cop came to her and he wanted a blood draw because he wanted to get a sample. I don't even know if he was looking for genetic material or if he was looking, I think he was actually looking for a blood alcohol content on the person. And she refused to draw it because she didn't have consent of the patient because he was unconscious, and the officer didn't have a warrant. And he argued with her and she stepped up to him and she ended up getting hauled out of the hospital. It was like a big whole thing.
She's like,
so yeah, your stuff that is part of your body is your stuff. It's private, it's your stuff. But yeah, if you had other people who definitely have a counter argument for it. So explain what the Genetics Information Non-Discrimination Act is. So it's discrimination based on genetic info. So basically saying if you know somebody is carrying a gene that's is genetically susceptible to Huntington's disease, you can't refuse to hire that person because they're going to develop Huntington's disease as they age, that kind of thing. So most GM plants are grown in the US. Growing GM crops requires fewer energy sources than growing traditional crops. With all the questions raised about genetically modified agriculture, the wider use of biotechnology has not been blocked, just keeps going. Federal laws in the US do not require that GM foods be labeled as such. This was a big deal 2 or 3 years ago. Should GMO foods have a label saying they are GMO foods? And there was a whole argument about whether or not they should be federally required to. GM foods are required to undergo safety testing before they enter the market. They're not required, not any more than anything else. This is your standard safety testing, but nothing beyond that. So our pros and cons of genetically modified foods, we've talked about these a lot so far. So one that we didn't mention though, is that seed patents. So there are smaller farmers who can get put out of business by large companies that hold patents for seeds. You'll probably hear about Monsanto if you ever watch any of these documentaries about genetically modified crops or whatever. Monsanto is a huge conglomerate company that basically has a stranglehold on a lot of the agriculture systems in this country. Like they have patents for the seeds and things like that. And a lot of people look at them as like a big evil thing. So Monsanto, you could look up Monsanto and read all kinds of interesting stuff. So some proponents of GM agriculture argue that GM crops are safer than others. And they're saying this because they haven't been sprayed with pesticides, they have added nutrients. So they are resistant to herbicides and they don't have to be careful about how much weed spray they use. So bioethics question, it's easy to move genes from one species to another. Is it right to do? So again, there's an affirmative and negative argument. Hey, so what I'm going to do now is put up for you your test topics, unit 4 test topics. They will. You will need to know about alleles, homozygous versus heterozygous, dominant versus recessive, haploid and diploid with somatic or autosome and gamete, chromosomal sex determination, sex-linked traits, meiosis and gamete production, multiple alleles, polygenic traits, molecular construction of DNA. Think back to the very beginning when we talked about how the structure of DNA was discovered and how certain things fit together a certain way because the way the molecules are organized. Know what kinds of molecules exist. I mean, what kinds of the shapes of the molecules and how they come together to make the construction of DNA. And you'll need to know about the discovery of the structure of DNA. Go back to that slideshow from that day. Remember in the end, who in the end got the structure of DNA right?
Watson.
Publicly, Watson and Crick. Where did they get their essential piece of information from? Rosalind Franklin.
Rosalind through the
Yes.
traitorous man.
Mhm. Know about transgenic organisms, prokaryotic and eukaryotic DNA. What's the difference between prokaryotic and eukaryotic DNA?
nuclear envelope.
Where it's located, correct. And also remember prokaryotic DNA is circular. So you're gonna have to give me an example of the genetic engineering, something we've talked about in class. We went over a bunch today. I'm sure you can remember some of them because they're downright weird. Mutations and mutagens, location of protein synthesis, karyotypes. Remember what karyotypes are? If you don't remember what they are, go back and look. Transcription and translation, codons, introns, exons, promoters. Do you remember what those
I
are?
remember what intron exon is.
Yep, it's there. Go and look. You'll see it. Operons. Sorry. Operons are groups of genes that work together. Put that back up for you. Okay, so there that is. I will tell Shannon later, Shannon. Bye, Shannon. So I will close that out for her. And then I'll put this, it's going to remain up.